All right, welcome back everybody to the great fun that we're having that we're calling microbiology. This is chapter 2. This is a review of chemistry concepts that hopefully this is a review for you guys, since you were supposed to have taken chemistry before you took this class. So it shouldn't be a whole lot of new information for you guys. It will be a lot of biochemistry type stuff, organic chemistry type stuff, and we actually do use quite a lot of it in this course. So if it's something that you're having a lot of difficulty with, you might want to go back and brush up on it. So let's get right into it. I always forget to click. All right, so these are our learning outcomes for this chapter. If you guys are interested, they are available here. All right, touching back on that concept of atoms, bonds, and molecules and what those are. Remember, matter is any tangible material that occupies space and has mass. An atom is something that has to make up matter. Matter is made up of atoms, right? So these are tiny particles that cannot be divided any further without losing their actual properties. So the properties of atoms are made up of protons, neutrons, and electrons. Of course, we know protons and neutrons are much larger than electrons. Neutrons do not really have a charge. They have a neutral charge. And then protons have a positive charge, while electrons have that negative charge to sort of balance them out. So you would have an equal number of protons and electrons in an atom in its typical, elemental state. This is a depiction of atomic structure, essentially looking at the nucleus in the center. And so in this case, we have hydrogen, where it has, I know that this can work the way that I want it to instead of messing up. Let's see. All right. Here's its nucleus. And so we would consider this to have, a proton hanging out here, and we have its little electron hanging out there, so 1 and 1, and some other examples given below. So this is our line drawing of that situation. And remember that the orbitals that are drawn out here, these lines, those are just depicting general regions of where the electrons could be found in that area at that level of where the electrons exist, and that they can even jump from orbital to orbital. So keep that in mind as well, having to deal with the energy of the electrons associated there. Moving on to elements, isotopes, and radioactive isotopes. So an element is an atom with a characteristic atomic structure and predictable chemical behavior. So this is going to be, zinc and hydrogen and phosphorus. These are actual elements. So these are not going to be dealing with molecules, which molecules are more than one, atom bound up to one another. So an element is just a single, atom with its own characteristic and atomic structure and predictable chemical behavior according to only that element. It's made up of nothing else. That's what that periodic table is based off of. All right, isotopes are variants of the same element that differ in the number of neutrons. And then radioactive isotopes, we're having unstable nuclei that can release energy in the form of radiation. This can be in the form of particles that are coming out of the atom, or other ways that waves of radiation can be released due to the energy associated with that instability of the atom. So often use these in medical research and stuff like that. So that is how the periodic table of elements is designed, based on those elements and their characteristics. When we look at something like the characteristics of oxygen versus the characteristics of, zirconium out here, very, very different from one another, right? And based on how these elements will behave typically in their natural environment as their pure form of these elements. That's how we've established whether they're solid or liquid or gas, or they're unknown. And then we have the metals, of course, the metals, and then some non-metals like the noble gases. So that's how that's all situated. A molecule, that term refers to any chemical substance that results from combining 2 or more atoms. So in the case of O2, the oxygen that we breathe, that oxygen, atmospheric oxygen. In the case of nitrogen, atmospheric nitrogen, both of those, and O2 or N2. being molecules made up of the atoms of those elements, right? So compound refers to a molecule that's made up of different elements. So now we are introducing another element into the, into the equation. So in the case of H2O, we have hydrogens and oxygens, for example. Right, there are three types of chemical bonds that you really need to be familiar with for this course. We are talking about the covalent, ionic, and the hydrogen bonds. Covalent bonds, we're going to get into talking about these a little bit in detail, but covalent bonds are going to be a sharing of electrons. Ionic bond is going to be a transfer of electrons from one atom to the other and kind of stay that way. And then we have hydrogen bonds, which we'll get into the details of that here in a second. So covalent bonds, these are, again, sharing of electrons from between two atoms. A single covalent bond can share a pair of electrons, so one from one atom and one from the other atom, right? And then, we can have more bonds added in there. I'm not too concerned about going into the details of that. Polarity of molecules, however, is determined by the electronegativity of the atoms that are involved. So if we're talking about a covalent bond between oxygen and hydrogen in a water molecule, which is what this picture is showing us here in this slide, oxygen is going to be more electronegative than hydrogen and is going to essentially hog or attract the electrons more than the hydrogen will. So that is going to form a polar molecule due to that effect. Is that also wanted to say about it? I think it is. So when you have non-polar molecules, obviously there's not going to be any electronegativity involved. So then we have ionic bonds. These are whenever electrons are transferred from one atom to another. We're not sharing our electrons in these cases, literally just moving the electron from one atom to another. And then that can result in ionization. So if we put our two ionically bonded atoms or molecules into a solution, a solvent, in this case, let's think of it as being water, because that's the most commonly, used solvent here. Then those two ions will disassociate from one another, and form, an ionized solution. So we have the one, like if we go back to the picture that we showed of this, our sodium atom is donating its electron onto the chlorine atom. Chlorine atom is gaining an electron, so it's being reduced, and then the sodium atom is losing an electron, so it's being oxidized. We'll come back to that later on, but I do want to start introducing that concept so it's not totally new when we do come back to it. So that's what's going on there with that exchange of the electron. But of course, now that we've lost that electron that was balancing out the charge from the nucleus where we have the protons that are positive, right? So now we have a positive charge to us when we gave up that one electron. So that'll get, just one plus at the top for the Na, and then the chlorine atom will get a negative at its little top for its negative charge for having that extra electron in comparison with the protons that are in the middle. So that's what's going on there. So those cations are the positively charged ions. I think of it as cation has a T in it. That's like a plus sign to me. And anion, those are going to be negatively charged ions. So chlorine would be the negative and the sodium would be the cation, right? So electrolytes are substances like salts, acids, and bases that release ions when dissolved in water, like salt. So sodium and chloride, but we were just looking at table salt. This is just showing you ionization in a solution whenever we have added in our ionically bonded sodium chloride into the water, and those sodium and chlorine ions are able to ionize due to the polarity of the water solution that they are, being dissolved in. That's all that interaction is going to be going on and interacting with the water molecules themselves due to their polarity. Hydrogen bonds are formed due to attractive forces between nearby atoms or molecules. So essentially you have polarity going on where we've given our hydrogens their slight positive charge, and there's a polarity going on where our oxygens have like negative charge, or nitrogens have charge as well that can be attracted to hydrogens. And then those associations from those hydrogens, which are being affected by the polarity of whatever, electronegative atom they're attached to, and, other atoms that have also been affected by their own electronegativity, those associations can, allow molecules to associate with one another without having any exchange of electrons. We're not sharing electrons here, and we're not donating any electrons here. We're just associating due to polarity, essentially. So it is a temporary bond and can be easily disrupted. However, a lot of our molecules, our macromolecules, like our DNA or our protein, anything like that, depend very heavily on hydrogen bonds. So if I'm telling you that they're weak, and I'm telling you that they can be, affected due to, polarity and electronegativity around them, then it should make sense to you whenever we talk about pH, that could very seriously affect, the interactions of molecules with one another whenever they depend so heavily on these weak hydrogen bonds. So that's something you need to keep in mind as well as we go through this chapter. Van der Waals forces are just weak attractions between molecules that have polarity, but these guys are going to remain associated. They're not going to be things that are very easily disrupted, but they're due to polarity as well. You don't really need to worry about Van der Waals forces as much. We're not really going to talk about those that much again. So again, hydrogen bonding in water, and very, very important. We have the polarity, that negativity of the oxygen, since it's electronegative. Hogging the electrons has a negative polarity, whereas the hydrogens now have positive polarity because they are having their electrons stolen away from them, so they tend to ease towards positive. And then the negatives and the positive can associate with one another and be happy and whatever. And if we introduce other molecules into this, they can, relate with all of that as well due to the polarity of the hydrogen and oxygen molecules. Okay. When talking about the chemical equations, so anytime we have any sort of a reaction going on, any sort of a change in a molecule at any point ever, then of course there's going to be a corresponding chemical equation that we would be writing it out as, right? We always have the reactants on the left and the products on the right. The reactants are the molecules that are going in, that are starting chemicals, and then the products are our resulting end product, right? We have different kinds of reactions, like synthesis reactions. We're going to put some things together to make, a combined unit. And then we have the decomposition reactions where we are breaking things apart, essentially. Catalysts are substances that increase the rate of the reaction. They themselves will not be altered or consumed, at least not permanently altered or consumed in the process. Enzymes are organic catalysts that work for a lot of the processes that are going to go on in cells. And we'll talk about that a little bit more when we get into the metabolism chapters. So anytime that we're talking about solutions and solutes and solvents and everything like that, we mentioned it a little bit in a few slides ago, but a solution is a mixture of one or more substance that cannot be separated back again due to filtration or settling. So they've been dissolved together. You can't take them apart unless you are removing one or the other, like through drying it out or something like that. So a solute is going to be the thing that is dissolved in the solvent. Okay, so I know because these words are very similar to one another, but do be familiar with which one is which, please. Solute, that is a substance that is uniformly dispersed within a solvent. A solvent is a dissolving medium like water. Water is the most common solvent in natural systems. And then concentration expresses the amount of the solute in a solvent. Okay, so how much sodium and chloride are present when we dissolve salt into as our solute into our solvent of water. So that's dealing with that. So we can determine concentration by weight, volume, or percentage, depending on whatever type of a thing you're talking about. So water has a polarity that's indisputable. We've already talked about that. That's the oxygen being negative and the hydrogens being positive. That polarity of water allows hydrogen bonds with other molecules, not just between water molecules, but other molecules that would be amenable to associating with slight negative or slight positive charges of other molecules. So ionic solvents will dissolve because the cations are attracted to the negative pole and the anions are attracted to the positive pole, just like we were talking about with the sodium chloride, right? The sodium positive, it's a cation, it's going to be associating with that negative oxygen aspect, right, of water molecules. And then the opposite would be the case with the chlorine. It has a negative, so it's an anion, so it's going to associate with that positive aspect of the hydrogen atoms of the water molecules. So you get the idea. So each ion will become hydrated associated with water molecules. There's some molecules that are going to be polar and will, associate well with water. Some molecules that will not have any polar aspect to them whatsoever, and then in a way are almost repelled by water. They don't associate well with water at all. Like when we have oil and water trying to mix, they don't want to mix. That's due to this situation. So hydrophilic, this is like table salt. These are things that want to want to associate with water molecules that are polar, right? Because they themselves are polar. So typically hydrophilic molecules are going to be also have some polar tendencies. Hydrophobic or water-fearing molecules, these guys are like your lipids and your oils and things like that, where they do not have any polar aspects to them. They are non-polar, so they don't associate well with water. So they actually repel water. And then we have some molecules that are amphipathic, which may have, one at one end and the other at the other end. So like a phospholipid, like it sounds like, has like a phosphate at one group. So this just will be our phosphate. So that's going to have a bunch of oxygens around it. But that's what this circle around it means. That's the whole phosphate group. And then this is carbons and hydrogens all the way down. So that's like a phospholipid. It's not perfectly drawn, but that's the idea of how it works and how it functions. So it's so many oxygens around this guy, right? So this is going to have a very negative charge to it. And this is non-polar. So that is what we would call amphipathic. We have a polar head and a non-polar tail here. And these guys are what make up our lipid bilayer and helps protect, keep everything inside the cell that's supposed to be in there and keep everything outside the cell that shouldn't be getting in. Okay, acidity and the pH scale. So first of all, acidity is anytime that we have excess hydrogen ions. Anytime you have something that's basic, we have excess hydroxyl ions. This has to do back with the water. Okay, so whenever we have equal amounts of hydrogen ions and hydroxyl ions, so you see how if you took this H and you took this OH and you put them together, what would you get? You'd have two H's and an O, right? I mean, yes, so that is literally what this is representing. The kind of dissociated aspect of these hydrogen ions or hydronium ions, depending on how you want to look at this, and then our hydroxyl ions. So our hydrogens and our hydroxyls, if we have more hydrogen ions, then they aren't going to be balanced out by the hydroxyl, right? So more hydrogen ions is going to be acidic, whereas if we have more hydroxyl ions, that's going to be a basic solution. So all based on water and its makeup and the concentration of hydrogen ions is how we determine what the pH of a solution is. So pH scale is used to rate or determine the degree or acidity or basicity. That's a weird word that I don't know if I like that. Alkalinity is probably better. Acidity or alkalinity, which is the same thing as basic, right? So pH below 7. So 7 is our neutral point. That's water, like plain equal amounts of H plus and OH negative. So equal amounts of hydrogen ions and hydroxyl ions. That's a pH of 7. Okay, anything below the pH of 7 is acidic. Now, you're probably sitting there, but you're saying, but wait, so if 7 is neutral, but we were talking about this is a measure of the concentration of hydrogen ions, which is what pH is. Yes, you are correct. And then you just told me that pH a low pH is acidic, but that's supposed to be, according to our, let me see, this previous statement, excess hydrogen ions, increased hydrogen ions. So why does that number work out that way? Well, this is why this chart that you're staring at, probably wishing you weren't looking at. So when you look here, we go from 1 up to 0.00000 10 to the negative 14th. So out 14 times. Okay. So 7 falls here. So that's 7. And that is a measure of our hydrogen ions that would be, occurring with regards to our hydroxyl ions, right? So this is interesting. So they're equal here, 7 and 7. Okay, that's why it's neutral. So the more hydrogen ions we have, again, this is the measure of the hydrogen ions over here. The more we have, this is an increase, right? So this is clearly less than this is. This is less. This is more, right? So that's an increase. But the way that we measure this is based on after the decimal point. So this down here was 10 to the negative 14th, right? We've already mentioned that. So up here we have 10 to the negative 1, or we have 10 to the 0, which is just 1. 10 to the 0 is 1, by the way. So this is a logarithmic scale. Now, I know you love hearing it, but this negative is referring to, what we have coming after our decimal point, but this number out here is basically what our pH is referring to. So 0 is 10 to the negative 0. 4 is 10 to the negative 4. A pH of 12 is 10 to the negative 12. So you can see now that if, 10 to, if I tell you it's a pH of 2, then your brain should be thinking that's a concentration of 10 to the negative 2. So that's 0.001, because that we've gone out the 2 decimal places. So basically what I'm trying to say here is that you can tell just by looking at that that's going to be a higher number than 10 to the negative 14 is. So that's how you can try to remember, okay, how many hydrogen ions have we got here? Is it more than this? Is it less than this? 7 is a neutral. You're just going to have to remember that. I have to just tell you guys that it's a 0 to 14 scale. 7 is in the middle. So keep that in mind, but that's how that's measured out. Okay. That's pH in a nutshell. So anything with pH below 7 is a higher hydrogen ion concentration. That's a lower pH. That's acidic. A pH above 7 is basic, so that's going to be higher hydroxyl or lower hydrogen ions. Okay, neutralization reactions are where we're going to be reacting with acids and bases to give off water and whatever salts might result from the reaction. So this is just using some less words to describe what I was trying to describe, but pH is going to measure the acid and base concentration of a solution. It refers to the concentration of hydrogen ions and is a logarithmic scale of that. So it is a base 10 and 10 to the negative whatever. So think of it that way. Never forget that it's 10 to the negative something. And this is just a pH scale where we're showing some things that you might encounter in life and where they might fall on all of this. Human blood tends to be about pH of 7.4. If you were to look at milk of magnesia, it has a 10.5. We have vinegar down here, but a 2.4. It's very acidic. And so just some examples of stuff you might see in your daily life. We talk about carbon as being one of the most important, atoms, molecules, elements, that's probably a better word for it. Carbon is being one of the most important elements with regards to life, right? And organic chemistry in particular teaches us about organic molecules being carbon and hydrogen based. So carbon hydrogen backbone. That's the idea, right? So any molecules that do not have a carbon hydrogen backbone are inorganic. If they are having a carbon hydrogen backbone, so carbon bound with other elements, that's going to be the organic molecules, and that's what our life molecules are made up of. And why carbon? Well, it has 4 electrons in its outer orbital, which is relatively relatively stable for itself, but also much happier with, the 4 other bonds that it can make, or double bonds or whatever, and very stable atom, very able to create very stable bonds with other molecules. So it's a very good solid framework for a lot of the molecules that our lives depend upon. Now, the functional groups that are important with regards to organic chemistry, I would be familiar with these guys if it helps you guys to make a flashcard, but this OH group is a hydroxyl group. And then we have the carboxyl group down here, or carboxylic acid. That's typical. So our amino acids, this is the acid associated with an amino acid. But guess what? Amino acids also have an amino group. Yeah, go figure, right? So our proteins have that, and our proteins have this, one at each end of our amino acids. And that's what kind of makes up an amino acid. And we'll show those in a moment. But those are very important. Those are probably the most important ones that I'd be aware of. And then next would be the phosphate group. It's important to know about a phosphate group, like we just mentioned. These occur quite frequently in biology, are very important in the structure of DNA, RNA, as well as our energy storing molecule ATP. I can't stress it enough. So phosphates are crucial to that. So be aware of that and see how many oxygens they have hanging out around those. And we've already mentioned about how oxygens tend to be electronegative. So they tend to have that negative charge. That's important to remember as we go through these. So the main macromolecules that are, or the main biochemicals that I would be aware of for this chapter, we have carbohydrates, lipids, proteins, and nucleic acid. Those are the big 4, and we're going to go through each of those here. These macromolecules are assembled from smaller subunits or building blocks, and that is going to be broken down and as part of what we're going to call polymerization. So that's a repeating subunit of something bound to itself and itself and itself and itself over and over again. Or, like if we have different kinds of amino acids, but it's still amino acid, amino acid, amino acid, but, that would still be considered, a polymer. A monomer is the single subunit, the single version of those. Okay. What makes it up? So for carbohydrates now. Saccharide refers to a sugar. So your monosaccharide, when we have a whole bunch of sugars linked together, a polysaccharide. If we have, then we can look at the monomer for that polymer. The monomer would be the monosaccharide. So you have to think of it in that terms as far as understanding the chemical structure of all of these, which it is important to understand it if you are wanting to get a bigger sense of how, biology works and how it serves all of us. So, right, so a lot of monosaccharides and an ose, right? So glucose, fructose, sucrose. No surprise there. That's not any sort of a secret. We have different ways that we can depict the structure of carbohydrates. I wouldn't worry about it too much, other than, I would know that if when you see these sorts of structures with the rings, and there may or may not be depicted off to the side, some of these alcohol groups, those are alcohol groups, these OH groups, hydroxyl groups. In this case, since they're on the end of this, that would be an alcohol type group. These guys are very common in sugars. You can see that we have quite a few of them there, and they can't even determine what kind of sugar we'd be talking about when we're talking about like DNA versus RNA. So we'll get into that. But here's an example down below where we have a polysaccharide, and on the next slide you can see the interacting bonds between, sugar molecule, all bound together, and they're all bound across from one another. This is what makes up cellulose, which is what cellulose is like fiber. Whenever you eat fiber, whenever you eat a plant and you are, they tell you about how you don't digest the fiber, but, that's what this is. Fiber is cellulose. Next, we have the lipids. These are like the fats, essentially. So anything that would not be soluble in water. These are non-polar typically. So they have long hydrocarbon tails. First, we'll start with the triglycerides. These are storage lipids. So these are the ones that are going to be storing up essentially energy. So we have extra food that we eat, and then the body turns it into triglycerides to store, so we can use it for energy later on when we don't have as much food to eat. or you just keep eating a lot of food and then it just keeps storing it up a lot, like I do, like, because I like food. So anyways, that's how that works. So there's a lot of energy stored up within those long tails of the triglycerides. Anytime that you break a bond, it releases energy. So stores the energy that way. This is showing the triglycerides and how they can be, how their different structures they can have. So you have these ones here. These are saturated. That just means hydrogens are everywhere. There's no double bonds in the structure. When you introduce a double bond, it gives a bend in the chain there. So this one is unsaturated to some degree. And then whatever you're talking about, like, so this is linolenic acid, I guess. And like, I'm not gonna make you guys memorize that, of course, but the idea here is that based on where the double bond is, you can name it, 8, 6, 5, unsaturated linolenic acid or something. That would tell you that carbon 6 had a double bond there or something. That's the idea. So I'm not going to get into that, but that's what that means if you guys are ever reading that on the back of like a nutritional label. So the fatty acids, here we're talking about saturated fatty acids versus the unsaturated fatty acids when we're talking about those double bonds versus the hydrogens. The double bonds, if you break the double bonds and put the hydrogens and replace to take up those spots on the carbons, that's a saturated fatty acid, no more double bonds. So phospholipids, we mentioned previously, a big, huge part of our membranes. They have 2 fatty acids. There's a glycerol as sort of like at the top of it that associates with the phosphate group. That's just sort of a structural aspect to it. And then the phosphate group at the top with its charge, its negative charge associated with it. So we have a hydrophilic region and a hydrophobic region. So this makes it amphipathic. So here is the picture of the lipid bilayer, and you can see the phospholipids here. Here we have the head, and then this brown here is the little tails that are coming off of it. So that's the phosphate head, and then the lipid tails, the hydrophobic tails, and the polar heads on the outside. And of course, tails want to be associated with each other because they don't want to have anything to do with usually the water-based outside or the water-based inside. So that's why it sets up the way that it does and creates a membrane. If we need to get in and out of the cell, then sometimes we need to have special channels that allow things in and out of the cell because they can't pass through this special, sort of chemical situation where we have the charge on the outside and the hydrophobic, water-faring nonpolar aspect in the middle. That helps protect our cells that way. Yeah, so that allows for the way that these molecules interact with one another, allows for the fluidity. So if we want some more structure to our membranes for us as, like mammals, as multicellular organisms, we have cholesterol, which is a steroid type molecule that gives us a little more rigidity in our cells. So that's kind of the idea of what that functions as. It also works in the same way for some kinds of bacteria, which I don't want to get into too much detail of those yet. We'll get into those when we get into the bacteria chapter. And then we have waxes, that some bacteria have these waxes that help protect them from the environment and makes them hardy. They survive, being cleaned a little bit more. So they live a lot more. And so that includes tuberculosis and leprosy. Both of those have a waxy coating. Next, we have the proteins. The proteins, we say that they're the shaper of life because they are the predominant organic molecules in cells. These are going to be our enzymes. These are going to be basically everything, every function that you have going on in your cell. Your genes, let's just start there. Your genes, your entire genome, your whole genome, every single gene in your genome ever. Your genes code for proteins. They don't code for anything else. They code for proteins. So everything about you in your genetic material is going to be spat out as a protein. Now, some of those proteins go on and, make non-protein molecules or associate with non-protein molecules, but still, that's what your genes code for. They code for proteins, and that's all. They don't code for anything else. So this is the molecule of life. This is what determines who you are and what you are, and all of this stuff. Well, not exactly, of course you have your life, but I just mean like genetically speaking, the things about you, genetically speaking. So, and that's true for our bacteria, and that's true for your dog and your cat and, you know, your horse and, you know, the cow that you ate and all of that. All of these organisms, their genetic makeup, they code for proteins. So when I say it's an important molecule in your life, I'm talking about the most important, probably. Of course, then you could argue that DNA is because it codes for it all. We can get into that later if you want to, but man, proteins, they do it all. They really do. So they determine structure, behavior, and unique qualities of organisms. That's like a watered down version of it. I feel like I'm not doing it justice with that explanation, but hey, it's the one you're getting. So amino acids are our building blocks. We keep talking about, you know, what everything is made up of, as far as our polymers being, you know, made up of all these monomers linked together. In the case of a protein is amino acids linked together. And I don't know if you guys know this, but there are 22 naturally occurring forms of amino acid. In us, in our bodies, we have 20 of those. So these are all linked together by what are called peptide bonds. And those are going to be bonds between that amino group, which was the NH2 group. And remember that carboxyl group or that carboxylic acid group, that's part makes it an amino acid. Those two. So when those link across to the next one, that bond there is the peptide bond. And so it's just that repeating over and over and over again. Here are the 20 amino acids that we can know. Of course, I'm not going to make you guys memorize this right now. I'm not going to make you guys memorize this, period. But these are what they are. If you probably heard of some of these and not even realize that they were amino acids, things like phenylalanine. So if you ever had like the gum or it said contains phenylalanine, you know, I don't know, warning to phenylketonuretics or whatever, that they can't metabolize that safely. And then we have, I don't know, there's a lot of aspartate and glutamate and glycine and histidine. I don't know. To me, these are all, and tryptophan. Of course, you guys have heard of tryptophan with eating turkey. These are all amino acids. And so these are the guys that are building blocks for all of the proteins that are going to run our cells, and that's your genes code for. So amino acids. They look like so. So you can look at them. They're a prettier picture out at the side, but if you want to look at that, that might not make sense. But this is the part I'm going to focus on first. This is our like kind of base structure, is the blue portion. And so this is our NH. I'm going to draw it out to the side so we can draw this accurately. So that's NH2. Doesn't really matter which one's in front or not for this, because it's not like completely structurally accurate or anything. And then we have kind of our middle guy. That's kind of our determining what determines if it's, you know, tryptophan or phenylalanine or whatever it is. And then we have our carboxylic acid or carboxyl group. This hydrogen here is what makes it an acid. That's what can be donated out as an acid in the, you know, the typical solution, whatever. But anyways, what I'm trying to say here is when we talk about peptide bonds, if we lose this, if we lose this guy, you know, and it's gone, and we, you know, dissociate from one of these hydrogens associated with this, then we can get a peptide bond. So we would have, you know, this H, and so this is NH2. I'm just drawing it out this time, see, and so on and so forth. And this would be your R group off of here. This is a really beautiful drawing. I'm sure this makes total sense to you guys. So anyways, that's the idea of how those link together. And then you can have that repeating and repeating and repeating and repeating over and over again. So the alpha carbon is the one that's in the middle that's associated with the R group. The R group, where I drew the R, is literally what's going to determine if it's whatever functional group goes there, whether it's just a hydrogen or it's this giant ring associated with it for phenylalanine or whatever it is that's associated with that R group, that's going to determine what amino acid it is. What else I want to say about it? So we've linked these guys together over and over and over again, amino acid and amino acid, amino acid, and those R groups determining what kind of amino acid it is. Now we can create a peptide. A peptide is a short chain of a few amino acids, but once we get more than 20, we are a polypeptide. It's now long enough for us to be thinking, hey, this could have some sort of a function. So not all polypeptides can be considered a protein. A protein is when it has its function and is able to perform its function. Sometimes polypeptides can do that. Sometimes they have to do other things first in order to do that. So either associate with other polypeptides, they come together as a whole bunch of them together, or have certain modifications added on, like a lipid group or a sugar or something like that added on to them in certain places. And either way, whenever you have your long chain in your polypeptide of your amino acid 1, amino acid 2, amino acid 3, amino acid 4, phenylalanine, tyrosine, tryptophan, and whatever, that is your primary structure. So that's just like your basic amino acid sequence of that protein or that polypeptide. The secondary structure arises whenever the functional groups on the outside will interact with one another. So now we start having some of these polar aspects interact with other polar aspects because they're being repelled by the non-polar ones. Whenever we start having folding going on due to those molecular interactions. This is where we start seeing the alpha helix, which is a coil, just what it sounds like. And then the beta pleated sheet, which is like, they usually draw it like an arrow, and then it'll usually curve around, and then you'll have, they call it a pleated sheet because it kind of looks like it, but it isn't necessarily. But it's not got any helixes in it. It's the long, smooth sections that kind of fold back and forth on one another. So that is the beta pleated sheet or accordion fold. So official name, beta pleated sheet. The other one with a helix that curls around, that's the alpha helix. These shapes occur solely due to the interaction of those R groups with each other and with their environment. The tertiary structure now, we've got our alpha helices, we've got our, beta pleated sheets, we've got whatever rando other things could be occurring. That's basically your main two structures are going to be your sheet and your helix. And there's just other stuff that happens too, because not everything's perfect, not always a helix or a sheet. So now we've got all these other structures interacting with one another. As we've got our helices interacting with other helices or with the sheets or everything. So these other structures that we've made start interacting with one another based on how they're folding, and also the formation of covalent disulfide bonds. Sulfur groups are hanging out as part of the R groups. So it might have part of it might have sulfur, which normally has a hydrogen, and then we have a sulfur, which normally has a hydrogen on another side. So we folded over, our protein is all folded over, and then we're interacting with one another. These sulfurs, a disulfide bond is whenever these sulfurs bond to one another covalently. So they're actually covalently bonded, sharing their electrons with one another. So that's what's going on there. That's the tertiary structure. That's wherever we have, I guess a bigger association on a larger scale as far as these molecular interactions based on polarity and non-polar regions and other aspects of these proteins. So then we have the quaternary structure. We might have to have, we make this big old blob, basically essentially this folded polypeptide chain. Due to our tertiary structure now, we might have to have several of them come together and work together in order to create the functional protein. So the protein and its ability to actually perform its function might not happen until we have several subunits come together and interact with one another. So it leads us to the concept of an enzyme. Again, an enzyme is a catalyst, and a catalyst is basically anything that helps drive a reaction without being used up, right? So catalysts, organic catalysts in the cell are referred to as enzymes. They usually have specific functions in specific areas. That all has to deal with the structure and how it's folded up and everything like that. So structure is incredibly important in a protein. A lot of that is determined by hydrogen bonds to one another. So whenever we go back to look at this picture and I'm telling you about the formation of these alpha helix and the beta sheets and these, the actual functional, what is happening? I don't want to quit. Okay. And sorry, I clicked a button apparently. So, right. So look at this. When you look at the polypeptide version, the actual, like, say, finished product, if you will, of the protein, and all of these things that are interacting with one another and how they came to be that shape and where the alpha helices might be, or how those alpha helices are interacting with other alpha helices and everything like that, that's determined by hydrogen bonds. A lot of that is going to be held together and associated with one another via hydrogen bonds. So this is why pH is so important in a biological system, because you would be disrupting this whole interaction if you disrupt the pH. That's why we learn about this. All right. Antibodies are a kind of protein. They are glycoprotein. Glyco just means there's the sugar groups on it. That's all it means. Nothing special there. So they're glycoproteins that have very, very specific shapes and very, very specific interactions with parts of bacteria, viruses, and microorganisms to allow our immune system to fight against bad invaders. The native state of a protein is the normal functional three-dimensional form of that protein. It's able to do its job, whereas the denatured form, there's been heat, there's been a massive change in pH, something like that, that has caused the hydrogen bonds to no longer be able to associate with one another, disrupts that whole thing, and the whole thing falls apart and can no longer function because the shape is not right. This is like when you cook your eggs and you can't get them back in the shell. You can't get them back how they were before you cooked them. That's denatured the proteins. Next, we have the nucleic acid. So this is going to be DNA and RNA. DNA is your genetic information. I call it like the blueprint, essentially. And then we have the RNA, which will serve as our copy, our working copy. So you're not going to take this blueprint out of like the archives of the Smithsonian. So you're not going to take this guy out of the Smithsonian. You're going to make a copy of the part you're interested in. And then you can take that out of the Smithsonian to, build your house to, mirror whatever that says, right? So that's the idea with this. So your DNA is your hard copy. Your RNA is the working copy. It has other functions for the cell as well. We'll get into that later. So they are made up of nucleotides. That's their, subunit. A nucleotide has a nitrogenous base, which is down on the bottom of this little guy down here in the orange and the yellow. So these are the nitrogenous bases. DNA and RNA have a nitrogenous base, a pentose, which is a 5-carbon sugar, and a phosphate. They both have that. They have all three of those. There's differences though. DNA has the nitrogenous base thymine in addition to these three, whereas RNA has the nitrogenous base uracil, but it does not have thymine. DNA does not have uracil. So you basically replace thymine with uracil for your RNA. The other difference is that RNA has a different sugar, which is why it's called what it is. So DNA is deoxyribonucleic acid, and then RNA is ribonucleic acid. Well, I ain't got the deoxy. That's really the only difference. And that's literally the difference there. There's sugar. When you look here, this is ribose. This is deoxyribose. You see why it's called deoxy now? Because we literally take the oxygen out. So that's what's happening there. They're similar sugars. It's just one of them doesn't have oxygen there. So it's deoxide. So deoxyribose, that's the 5-carbon sugar. Deoxyribose and ribose, 5-carbon sugar, the pentose. Then our nitrogenous bases, they both have adenine, cytosine, and guanosine. Guanine, A, C, and G. DNA has the T, RNA has the U. And we'll remind you about this when we get into the genetics of these. I hate this part, talking about purines and pyrimidines. It's just, I can never remember them, which one is which, and I'm probably going to have you guys remember it, which sucks. But if you remember for one test, that's probably all you need to know for. So our purines are 2 rings, essentially, for our nitrogenous bases. This includes adenine and guanine, whereas the pyrimidines, these are our nitrogenous bases that have 1 ring, and that includes thymine and uracil, because they're pretty interchangeable, and then cytosine. So those are our purines and pyrimidines, and those are the kinds of nitrogenous bases that we can have. Of course, the 5-carbon sugars can either be deoxyribose or ribose. So those are the differences. We already talked about that a little bit, and then this is just showing us the helix, the double helix for DNA, and how it interacts with one another. So the nitrogenous bases form bonds across one another. A always bonds with T or U in the case of RNA. And G always bonds with C across when we're having this. So this is their little matchup across their little helix. We'll talk about that again more in the genetics chapter. So RNA, again, our working copy, right? I've said that we have our DNA as our genes that we're not going to take out of our Smithsonian, which is our nucleus. We make a copy of it. That's our RNA. So it's our messenger RNA. It's carrying the message of whatever gene you're interested in. We can take that out of the nucleus and go work with it where our workhorses are. So that's our ribosomes. Ribosomes are going to read the messenger RNA. And the way that they do that is they use transport RNA, tRNA, to carry amino acids based on how they match up with the messenger RNA. And then the ribosomal RNA makes up the actual structure, most of the structure of the ribosome itself, that's going to be doing a lot of the reading of the messenger. So that's how that's going to work. Again, we'll talk about that more in the genetics chapter. So the energy molecule of the cells, this is going to be ATP. ATP is adenosine triphosphate. So we're going to have our adenine group. It's our nitrogenous base. We're going to have our ribose sugar, and then we're going to have 3 phosphate groups. Okay, so adenine, nitrogenous base. It just means there's nitrogens in it. It's not a secret or anything. Ribose, this is our pentose sugar. And then our 3 phosphates. So we've talked already about how phosphates have that oxygens all around it, gives them that negative charge. Okay. So this is our energy molecule. I don't want to give it away too much yet because we're going to talk about this a lot in the metabolism chapter, and also have our end of chapter question that has to deal with this, FYI. But so you see all these negative guys near one another. So energy is going to be released whenever we break the bond between the 2nd and 3rd phosphate. So, at the end, clip the end one off there. That's how the energy is stored. So ATP, the triphosphate, storing the energy, we clip off the phosphate group, releases energy, and now we're left with ADP, adenosine diphosphate. And then we can go add another phosphate onto that again through other processes like aerobic respiration, which we'll talk about in our metabolism chapter. So cells, this brings us to that concept of cells. We talked about all the building blocks of cells essentially so far in the chemistry chapter. As far as the chemistry of cells go, we have a lot of carbon, hydrogen, oxygen, nitrogen, and other atoms. And we'll get into the details of that. This follows the basic laws of chemistry and physics. These are our fundamental units of life. They produce characteristics, reactions, and products that can only be described as living. Viruses do not have this. Bacteria and protozoa consist of only one single cell. They're not a multicellular organism. A multicellular organism, you take the cell off the organism and it can't live without the organism. Single-celled organisms can live on their own. So there's different shapes of cells, there's different functions of cells, different sizes of cells. Just depends on what they're being used for and what organism they're involved in. Eukaryotic cells, these are, remember, these are going to be our cells that have a true nucleus and true organelle. So they are membrane bound. So the membranes that we just talked about with the phospholipids and the phosphates on the outside and the hydrophobic tails on the inside of them, that's a bilayer. Just FYI, that's a lipid bilayer. And those cells are enclosed. Well, bacteria and archaea have that too, but the difference is that we also have more on the inside that also are containing our nucleus, also containing, you know, our mitochondria, which are our powerhouses of the cell, and things like that. So that is the idea of the eukaryotic cells. They're a little bit larger. They tend to be a larger cell. We've got membrane-bound organelles. So those are the things that are going to be making things like energy or whatever. They have a function. Whereas bacteria and archaea just have everything kind of going out in the cytoplasm or the goo inside of their cells. So that they are, like it says, very structurally complex as far as the cells are concerned, because they have to have a lot going on in their cells because they are single cellular organisms. They can't get help from other cells in their bodies like we do. So that concludes chapter 2, the chemistry chapter. The question of this chapter is, what do we do about chemistry and how molecules, knowing what we do about how chemistry and how molecules interact with one another. Looking at ATP, that energy molecule, why do you think it makes a good energy storing molecule? Why do you think it is able to serve as an energy storing molecule for our cells? So I'll leave you guys with that thought, and I will see you guys in the next chapter. Bye-bye.